My name is Elizabeth Wasmuth. I am tenure track assistant professor at the Department of Biochemistry and Structural Biology at the UTHealth San Antonio. Our facility just opened in July. Currently, we have one microscope, a 200 kV Glacios, equipped with a state-of-the-art detector, a Falcon 4, and it is equipped with a Selectris energy filter. No one would have thought it a year ago, and even right before on the eve of the facility opening, people were skeptical, but we are getting 2.2 angstrom structures of previously unknown complexes. The technology, the Selectris, it works. It is really helping us achieve resolutions at 200 kV that we would have thought would have only been possible with a Krios. So we have this great ongoing relationship with Thermo. They helped us with the installation. They're helping us now with consultation and troubleshooting. And we're excited to keep that relationship strong so that we can see where all of the technologies will take us in the future. The wonderful part about structural biology, and it's almost an endorphin release, is the discovery of what was previously unknown. And so every time I've seen a structure of my proteins, whether or not it's by X-ray crystallography, but most excitingly cryo-EM, because you can see so many different shapes, there's just a thrill that runs through my body. And that's an addiction that keeps me up at night. What does this thing look like? What's the shape? How does that inform the function in a normal cell? How does that lead to prostate cancer? Even now, as we speak today, we're collecting on something that I never thought it would form this shape, and it's nuts. And cryo-EM can give you that feedback almost instantaneously.